Hello scholars, welcome back to another episode of Time-Based Guide to Not Effing Up A-Level Biology. Today we're going to be doing synapses and I'm pretty much going to go over all you need to know with respect to that. So the first thing we're going to do is start off by drawing a diagram, and you know how much I love diagrams. And synapses, I just really like drawing them out. I feel like they're this majestic counterpart of biology we don't appreciate enough. So yeah, this is kind of something I really like. So here we have synaptic knob. I'm going to draw out the actual structure of it. I'm going to explain later what each part does when we go over how an action potential passes from one neuron to the other. But let's start easy. I'm going to draw out a few of these channels, and these are called your calcium channels. I'm going to draw out some mitochondria here and there. Mitochondria. I'm going to draw out some, for funsies, let's do a few ERs here and there. I know, ugly drawing. We have a few ERs, and finally I'm going to draw out a few vesicles, and they're going to have a neurotransmitter in them. Now, the neurotransmitter we need to know about is acetylcholine. But you can have different types of neurotransmitters like GABA or dopamine or serotonin. There's just so much like different neurotransmitters, but for the spec and for how nerve impulses will propagate in this section, we're specifically going to be looking at acetylcholine. So vesicles containing acetylcholine. There we go. So that's the general structure of a neuron. Obviously there's a nucleus and other stuff, but that's on the other end of the neuron. So that's what we have here. I'm going to draw out a bit of the other part, so the other end, effectively, of another neuron. And I'm just going to be labeling what we call these parts in particular. So this part here is called your synaptic cleft. This is the presynaptic bulb. And you can just call this the postsynaptic neuron. I think there is another word that's a bit better for it. Oh, okay. The postsynaptic cleft. So I guess we can call it that then. I mean, you can call it neuron if you're more specific, cleft. There we go. So just so that you know, kind of like what we're referring to when we're going over this topic. We're going to go over summation quickly, so this shouldn't take too long. Summation is effectively this idea that, when we were talking about action potentials and the fact that you need to reach a threshold in order for an action potential to be released. So sometimes what we can have is that we can have multiple neurons sending out impulses, and those impulses can sum up, hence the name summation, but they can add up, and eventually they can lead to that threshold being reached, so we can actually release an action potential now. So there's two types of summation. There's spatial summation, and this is effectively where multiple neurons come together in a junction, and they release and they effectively stimulate an action potential. I feel like I should have went over this earlier, but a neurotransmitter is a chemical that is released from a neuron, and that chemical effectively causes an action potential to be stimulated at the next neuron. So with spatial summation, I'm just going to draw a little diagram here. So this is the postsynaptic neuron, and here is a few presynaptic, I know ugly, but like a few presynaptic bulbs over here, and they're all going to be releasing a neurotransmitter like acetylcholine. So if there's a few, we're not going to be able to stimulate a strong enough generator potential to stimulate an action potential in the next neuron. But if we have multiple of them sending out these really weak impulses, then eventually we'll have enough to stimulate an action potential. Now, the graph that you need to know, it kind of looks like this. So if this is like your action potential, it'll look a bit like this. So you have multiple small impulses, very discrete, and they're all releasing a bit of neurotransmitter, and they're adding up so that the neurotransmitter is at a high enough concentration to stimulate an action potential. The other one is temporal. And temporal is effectively, it's going to be very similar to this. So we have a postsynaptic cleft over there and a presynaptic bulb. It's just one ending. And what happens is that if there's multiple frequent impulses, then we're going to be releasing more and more acetylcholine over and over again. And there's going to be enough acetylcholine on the postsynaptic cleft in order to stimulate an action potential. And the graph of that kind of looks like this. It's kind of jumping. With spatial, the way I like to remember it is it takes up a lot of space. So you have all of these neurons coming together, and that's spatial. And then you have temporal, and out of the process of elimination, that has to be the only other option. So these are the two graphs you need to know. With spatial, small little jumps until we reach an action potential. With temporal, it keeps on adding up. There we go. Alright, so that's like your kind of, I don't want to say anatomy, but like cell structure kind of thing. The next thing we're going to do is actually go over the mechanism on how it works. So, yeah, let's do it. I'm going to do a few diagrams on the right hand side, just so that you can visualize what's effectively going on, really. There we go. I need to get a better sponge. Step 1. An action potential reaches the presynaptic neuron. I'm going to put action potentials as AP. An action potential reaches a postsynaptic neuron. And I'm just gonna, I don't think you really need a diagram for that if we've done the lesson on action potentials. So it reaches the postsynaptic neuron. What that causes is that action potential stimulates voltage-gated calcium channels to open. So I'm going to do stage 2. This causes voltage-gated calcium channels to open. So little bulbous, the bulbous head of the presynaptic bulb. And over here we have a bunch of these calcium channels opening. Now what you need to know is that outside of this bulbous head we will have a high concentration of calcium. So the next step is that this causes calcium ions from the outside of the presynaptic bulb to diffuse into the postsynaptic bulb. So I'm just going to depict that over here. So I'm just going to put a few arrows just to show that it's entering. Now, the reason why calcium is so important in synapses is because calcium ions effectively help release acetylcholine. So if I draw the next diagram over here, we have these vesicles of acetylcholine. I'm just going to put a neurotransmitter in them. And the calcium causes the vesicles to bind to the membrane of the presynaptic bulb, and it effectively causes the release of acetylcholine because that membrane will fuse with the presynaptic bulb and release that neurotransmitter. The actual process of this reaching, like fusing with the membrane, it starts off with a process called invagination. Don't laugh. And you can effectively summarize that as exocytosis. So let's say calcium causes vesicles containing acetylcholine, I'm going to put ACH, to fuse with the membrane of presynaptic bulb and release the acetylcholine to the synaptic cleft. This is an example of exocytosis. A bit of extra information doesn't help. Doesn't harm. There we go. This is an example of exocytosis. All right. So all of that calcium is starting to diffuse in. I'm gonna draw out a bit of a vesicle, kind of like fusing, and we're releasing that acetylcholine into the synaptic cleft. So stage 5, the acetylcholine diffuses along the synaptic cleft, and the acetylcholine is complementary to a receptor on a sodium channel. So on the postsynaptic cleft, we have, I'm going to do another color, I'm going to do yellow. We have a bunch of sodium channels, and the acetylcholine, it's complementary to a receptor on that sodium channel that causes that sodium channel to open up, and it will cause sodium to diffuse from outside of the postsynaptic bulb, postsynaptic cleft, sorry, and it will diffuse in to start stimulating an action potential. So let's put that into words. So where were we? There we are. Okay. The acetylcholine diffuses along the synaptic cleft and binds to receptors on sodium channels. These receptors are complementary, that's a key word, I'll highlight that in a bit, complementary to the acetylcholine molecule, let's say. There we go. So let me draw another diagram here. I'm just going to box these diagrams so it's a bit more evident. So I'll move over here. Here we have the postsynaptic cleft, sodium channels on the outside, sodium channels here and there that I've opened up because of that that acetylcholine. So I'm just going to put ACH here. And that sodium will start diffusing in. Now, this is starting to look a bit familiar because when sodium diffuses in, we're starting to depolarize the membrane. So we're starting an action potential here. So influx of Na plus causes depolarization of the postsynaptic. How would I describe it? Postsynaptic. Sorry. I just say the next synapse or the next neuron. Oh, there we go. Postsynaptic neuron. There we go. Which, when threshold is reached, remember about summation, it's not always going to happen. Remember, we have the all or nothing principle. It has to reach threshold or we're not going to release an action potential. So depolarization of the postsynaptic neuron, which when threshold is reached, fires an action potential. Now, there's one more thing that we need to know, and that thing is acetylcholine esterase. Now, enzymes often end in A's, so this should give you some idea of what happens. They're located on the postsynaptic cleft. I'm going to draw them in green. And what they do is that they hydrolyze the acetylcholine back to two parts. Acetylcholine is made of two compounds called acetyl and choline. And what it does, it hydrolyzes it so it can diffuse from a high concentration here back into the presynaptic bulb, and that acetylcholine can be recycled and used again. So next step is acetylcholine esterase, long name. I'm going to just say ACHE. Acetylcholine esterase hydrolyzes the acetylcholine into acetyl and choline. ACHE is found on the postsynaptic cleft. Alright. I'm just going to make sure. Alright, that's fine. So that diffuses back in. The final point is that, so yeah, diffuses from high to low concentration back into the presynaptic bulb. And here we use ATP to reform the acetylcholine from acetyl and choline. Now we can say ATP used to reform ACH from acetyl plus choline, and then stored in those vesicles. Vesicles, recycled. Can be used over and over again. So let's just go over this one more time. An action potential reaches a postsynaptic, presynaptic neuron. Apologies for that. Presynaptic neuron. This causes voltage-gated calcium channels to open. This causes calcium ions to diffuse from outside, from the outside of the presynaptic neuron to diffuse into the presynaptic bulb. Calcium causes vesicles containing acetylcholine to fuse with the membrane of the presynaptic bulb. You see, I'm trying to be as accurate as I can, like really precise, because the mark schemes are really wishy-washy and they want it to be quite accurate, so you don't want to be really vague with your terminology here. So calcium ions causes vesicles containing acetylcholine to fuse with the membrane of the presynaptic bulb, postsynaptic bulb. Again, another mistake, so don't make these on the exam, guys. Postsynaptic bulb, and the acetylcholine reaches the synaptic cleft. This is an example of exocytosis, a bit of extra information. The acetylcholine diffuses away from the synaptic cleft and binds to receptors on Na plus channels. These receptors are complementary to the acetylcholine molecule. I would also like to say that you want to say that these Na plus channels are in the postsynaptic cleft membrane. Again, being as precise as we can. Influx of sodium causes depolarization of the postsynaptic neuron, which, when threshold is reached, fires an action potential. Acetylcholine esterase hydrolyzes acetylcholine into acetylcholine. Acetylcholine esterase is found in the postsynaptic cleft. And then the acetylcholine diffuses from high to low concentration back into the presynaptic bulb. ATP is used to reform the acetylcholine from acetylcholine, and that can effectively mean that it's being recycled. Alright, complex, confusing, a bit weird, so my videos are just here to help you kind of, but I'd suggest looking at the textbooks. So textbooks have some pretty good information and detail on nerve transmission itself. Also watch a bit of Miss Estrick, pretty helpful stuff. Alright, so next lesson we're going to be doing actin and myosin, so hope that helped, and I'll see you guys in the next video.